Good morning, everybody. I'm very happy to be here, and I see at least one familiar face in the group. I thought I would start with some pictures, and I could just give you my Facebook account, except that I don't have many pictures there. So this is it. This is me. I am in biological and agriculture engineering. I'm married to a colleague right here, and we have two kids, beautiful, smart kids. It does snow in Louisiana, by the way. That happened a few years ago, and I wanted to share with you all that happens. Um, we have two professionals in our family, right? I'm married to a professor. I'm a professor. We have kids. How do we do that? Some of you might be asking yourself that. We bring in grandmas, so they help a lot with the kids and balance things out at home. But I have two at home, two kids at home, and lots at work. We really like to think of our groups as our families. We are at work a lot, so we should develop good relationship with the students. And I had very good relationships with my advisors, so I hope I can provide the same to my students. So this is very important in selecting your advisors. I guess you should know who they are and whether you can work well with them. And if we're looking at where I came from, you had a speaker before me from France, and I'm from Romania. So this is your European morning, I guess. And this is not changing. Now it will. Okay, so where did I come from? I said Romania. I have a bachelor in food technology. What is that? Well, it's very similar here. You do it in biological engineering as a food engineering major. But I graduated in 97. Before I was even done, I was recruited to come on an exchange student program to Missouri, and that was in my second year of college. And I absolutely loved it in the US. I decided this is it for me, I'm gonna come back. So I went back to Romania to finish my bachelor degree, but immediately returned to pursue a master's in agriculture engineering. And then I continued in North Carolina State University, where I got a few degrees, one in chemical engineering and another in biological engineering and another in food science. And I thought that by collecting degrees, I just learn more and I'm more competitive later on, because I didn't really figure out what did I want to do with my life. Now everybody tells me that I'll be a great professor, just at home, and my professors, my advisors really recommended that I should pursue a career in academia. And I think they were right, because I really, really love it, and I'm not going to quit it unless I get kicked out of LSU. I really like what I'm doing, and I'll be here for a while. So let me switch to my research. We're working on polymeric nanoparticles to improve health, and this stemmed from my interest in food and health. So we talked about cardiovascular diseases. I'm looking at cardiovascular diseases from a completely different perspective, and that is, how does food affect that? And are there any bioactives that could perhaps prevent cardiovascular diseases? You are what you eat, so that's important. But some of these functional foods that incorporate bioactives require a lot of expertise, because bioactives, vitamins, antioxidants are really great, and some have really great benefits for health. But the amounts, the bioavailability of those bioactives are really small. So can we use polymeric nanoparticles to improve bioavailability of bioactives, prevent disease, and improve health? That's the idea. So when I'm looking at nanotechnology definition, maybe you are exposed to this, you have been in a program, I'm not sure, but this is what I wanted you to look at. Nanotechnology is the understanding and control of matter at dimensions between approximately 1 and 100 nanometers, where unique phenomena enable novel applications. So what I have in red is what I believe is most important. Size 1 to 100 is important. but it's not critical. Small size is important. But understanding and control of matter is most important to me. And the fact that unique phenomena enable novel applications. Because I'm coming from engineering, from food science, I really am looking at the application part of things. So NNI, which is National Nanotechnology Initiative, has a vision of a future in which the ability to understand and control matter on the nanoscale leads to a revolution in technology and industry that benefits society. So that really moves me. And you need to be in a research area that really makes you tick and you care for. So I think that with nanotechnology, we have the potential to transform and revolutionize multiple technologies and industries, including some of the stuff I'm talking about, agriculture, food, health. There are different types of particles that you could use for drug delivery. Many types of particles. You can look at inorganic nanoparticles, polymeric nanoparticles, solid lipid nanoparticles, liposomes, you may have heard about those, nanocrystals, nanotubes, and dendrimers. To me, if you're interested in food, health, and delivery systems, you have to consider what are the health implications and the toxicity, the safety associated with the delivery system if you think about ingesting that particle. So with that in mind, we're thinking polymeric nanoparticles should be the way to go. We worked on combinations of inorganic and organic nanoparticles, but really now we're completely focused on polymeric nanoparticles because you can make them for biocompatible, biodegradable polymers, and you don't have to worry to some extent about their toxicity. There will be perhaps still some, but we'll talk about that later. So there are different types of polymeric nanoparticles that you can make and different things you can do to them to reach your objectives. You can entrap this drug in red inside the particle. You can attach targeting ligands on the surface to take the particle to the site of action if that's what you desire to do. So you can make polymerosomes, polymeric nanoparticles, nanoshells, dendrimers, micelles, and polymer drug conjugates. If you look at that, that's huge opportunity. You can do whatever you want. You're an engineer, I am, and you want to apply it to a health problem, to solve a problem. So you can design it working in a team of engineers, chemists, biological science people to create a system that fits your needs. So this slide shows that you have the ability to create a polymeric delivery system that fits your needs, whether it is a polymerosome or simply a polymer drug conjugate, depending on the application that you're interested in. This is what we do in our lab. And I am changing the size to 1 to 1,000 because, to me, if we're looking at submicron and we still find the novel phenomena, the unique phenomena, we're still in nanotechnology. So polymeric systems have the advantage that I already mentioned. You can make them biocompatible, biodegradable. You can protect the drug or the bioactive that you entrap if it's labile inside the particle. You can target, as I was saying, you can target the particle to your site of action. And very importantly, in my lab, we looked at enhanced bioavailability of vitamins and antioxidants. So because these particles are very small, you have a very good uptake and improved bioavailability of what you entrap inside the particle. And also, if you design it to have specific size and loading, different types of molecular weight polymer can be used, you can really modulate the release to match your desired outcome. So lots of different advantages. But my point is, you cannot take advantage of all the improved protection, control release, targeting, and bioavailability unless you go through in your research these steps. You have in the first place to be able to make particles of defined properties, very well-controlled properties, meaning morphology, size distribution, all those things are important. You need to, well, after we make the particles of defined properties, we look at their function. Is the stability of the antioxidant what we want? What are the release kinetics? What's the antioxidant activity? What's the specificity of the particles we make? And then this is where I hire a lot of students to undergraduate students to look at the bio-interaction. So now that we made particles of control properties, how do they interact with cells? C. elegans, and we take it all the way to rats. So we want to see what is the absorption, the biodistribution of the particles, where do they go in the body, and very importantly, do they pose any safety to us as consumers. So all those things we're doing, and I'm looking at research, and if you only focus on this part, but you forget in your paper to specify what type of particles you worked with, you just buy them from the web, and you say, I tested this in cells and they're safe, and you find great results here. But if you don't convey what type of particles you worked with, that type of research doesn't help the community much, because we don't know what you worked with. So you really have to start here, and then hopefully in my lab take it all the way to safety of particles. So I think as a requirement to work in my lab, you need to like to work at interfaces, because there is a little bit of chemistry, a little bit of engineering, a little bit of bio, and you should be able to work with teams, and those people are very successful. A little bit of chemistry doesn't hurt anybody. We're looking at synthesis of particles, and there are different ways to make these particles, as you see here, depending on what kind of bioactive or what kind of drug you put. You can use lipophilic solvent, an amphiphilic solvent, a hydrophilic solvent. And if we're just looking at the top part, you would make an emulsion from the solvent in which you dissolve the polymer and the drug, and then the water phase with the surfactant. You make that emulsion, and you try to make the size as small as possible, so you apply homogenization, microfluidization, sonication to make those emulsion droplets very small. And then after you evaporate the organic solvent, you end up with the particles with entrapped drug. Now, there are different steps that you may want to take further to perhaps target that particle, but this is really the basic step to make polymeric nanoparticles. So when we're looking at emulsion evaporation, which is that exact method that I just described, this is a more visual way to show what is involved. So you mix the organic solvent with the polymer and bioactive with the water phase, you homogenize, sonicate, and then evaporate the organic solvent to make the particles. But I said, particles have specific properties which will affect how they will interact with cells, where will they go in the body, and how toxic they'll be. So there are ways to really control those properties: size and size distribution, surface properties, stability, functionality. And that is simply by changing the types of materials you use, by selecting the right polymers and surfactants. Also looking at what concentrations, what ratios you use between the water and organic phase, and also at what pressure you use if you apply homogenization, or how long you sonicate, and some engineering parameters that you can change to control the properties of the particles. So after you make them, we're looking at different ways to characterize those particles. And we come to life sciences, to chemistry, and use transmission electron microscopy, SEM, FTIR, HPLC, confocal microscopes, to see what are the characteristics of these particles and confirm that we've met our design goal. The reason why particle properties are important is because those properties will dictate how they will interact with cells. Whether they are big or small, it will make a difference. You have big particles here not being endocytosed, and then you have some small particles that are loaded with either a hydrophilic or a hydrophobic drug. And after the particles are endocytosed, then you can modulate where that bioactive will go in the cell, and maybe the function of the bioactive will change depending on its localization inside the cell. Also, the properties affect not only particle-cell interaction, but where would the particles go in the body. So if you are looking at a very simplistic uncoated particles, that particle, whether you ingest it and it's getting absorbed into the plasma or you inject it, in the end, it will interact with the human plasma and many proteins will bound to the surface. So those particles will actually end up mostly in the liver and kidney. And they don't go through circulation and deliver the drug where it's needed. Well, if you pegylate it, you put a polymer polyethylene glycol on the surface, then a small amount of proteins are bound to the particle so that you can reach your goal of circulation and you can take the drug to where it's needed. And if you are interested in treating Alzheimer and Parkinson's and you are focused on delivery to the brain, then you can do things to those particles. Lately, we find out that if we coat polysorbate on particles, then you can bind up ApoE on the surface of the particles, and the particles will reach the brain. So my point is, you can be in the lab designing the particle to do what you want it to do, to deliver what you want delivered, where it needs to be delivered, at the quantities you want delivered, by just thinking how does that particle interact in the body. It's not easy. It's not easy to foresee that. So mostly what we are working on in our lab is intestinal uptake, and with the goal of improving bioavailability of vitamins and antioxidants. And what we developed are two types of particles shown here. A polylactic glycolic acid particle, about 100 nanometer in size, that is negatively charged at pH of 7. And then you coat that particle with a positive polymer, chitosan, to make a bigger particle positively charged. And we're thinking that these two particles will interact differently with the intestine and with the enterocytes, and I'll show you a movie to support our hypothesis. So what we're thinking is that if we ingest vitamins, those are packaged in these micelles by the bile salts, and some of the micelles will be absorbed through the enterocytes and made available, some are lost. If we entrap this vitamin in a big polymeric particle that is degradable and that adheres to the mucosa on the surface of the enterocytes, so we have a positively charged particle that releases the vitamin, then maybe perhaps we can improve the uptake of that vitamin, and the polymeric particle will degrade and it will be removed from the body, posing no safety issues to our health. The alternative is to take that vitamin, drug, bioactive, whatever you want to call it, and package it into small particles. These particles don't have the ability to adhere to the enterocytes, but because they are so small, the uptake will be greatly increased, and the particles will go to the blood and take the load with them. So now the efficacy of that transfer is much higher. You improve much better the bioavailability, but you have to worry more about the safety of the particle itself, because it may reach distant sites in the body where it's not supposed to be. And the particle made of polylactic glycolic acid is biodegradable, but if that particle accumulates, it takes a while for the polymer to degrade. So if that particle accumulates, say, in the liver for a month, that you may find some toxicological issues that we were looking to avoid. But different types of particles interact differently with the body, and by designing them specifically for your application, you can improve this bioavailability of what you entrap. And testing the toxicity, the safety of the particle is very important before we move on to commercialization. So the type of work that we do can be divided into these control properties. Some students really prefer to be in the lab making particles and controlling their size, characterizing fully the particle properties. By looking at different processing conditions, different types of materials, different types of uh, methods to make particles. We're labeling these particles, especially when we want to see where the particles go. We have to fluorescently label the particles. So you can use a regular fluorescent probe and physically entrap it in the particle to make it fluorescent, just like you would entrap the drug. But lately it's more common and more, I guess, scientifically correct to tag covalently this probe to the polymeric chain, so that when you find it in the cells or you find it in the body, nobody can argue that's just the probe that has reached the particle. So you can do that. We're targeting particles. We are doing it either to take drugs to the brain for Parkinson's disease treatment, or to kill termites. So there are different types of applications that you can think of. We're looking at functionality, the stability, release kinetics, antioxidant activity, specificity. I didn't show much data today, and maybe I should. But we have looked at fluorescent particles, and we did some ex vivo studies and in vivo studies to see where the particles go in a mouse. And also, we're in the process of looking at nanoparticle cell interaction and what is the safety of these particles when used in an animal model. So with that, I reached my background again. So I'll stop here and I'll take any questions that you may have. Thank you.
Is there a way to make the particles RNase free?
We the particles themselves, the synthesis itself involves the use of materials that I mentioned, a polymer, a surfactant. So if those are free, then you should be able to, if all the components are free and you work under those conditions, you should be able to. And particles are lately used very much in RNA delivery and DNA delivery as well.
Yeah, because the siRNA had major delivery problems were causing it.
Exactly. So you can solve some of that with particles if you can protect. We worked a little bit on DNA plasmid delivery, and we showed that with these chitosan particles, we can protect DNA from degradation, from DNases. But we haven't worked with RNA.
And in the vitamin video, what would make the small particles bind to the cell lining better than the body's particles?
Okay, so the way the particles interact with the mucosa is a function of their surface properties. And you can look at targeting specific cells, or you can simply change the charge of the particle, and you can assume that by making the particle positively charged, it will interact with a negatively charged mucosa, and you'll have some type of electrostatic interaction, which will make them adhere, and if they're big enough, stay there. What we found is that if they are positive and small, then you can really improve uptake by making them positive. But while working with the food industry, they really are not the pharma industry. They are really, really worried about the toxic effects of this toxicity of the particles. So some of them voiced this concern and said we'd rather have the particles big and stay there and release the bioactive rather than have the whole particle shuttle the bioactive because we then have to worry and prove FDA that particles are safe. Sure.
You spoke of injections and the particles entering the bloodstream. Have allergic reactions been a concern?
Very good question. Yes, we are looking at immune responses. And you can have immune responses to these foreign particles, obviously. And there are ways to go around it. And that regulating the surface of the particle is one way to avoid the macrophages and kind of decrease that effect. But every time you have a foreign particle, you have to worry about immune response.
I was wondering what they're made up of. Like, I'm working with carbon nanotubes. I wasn't sure if there was specific elements you were working with.
Sure. We're working with polymers. So this polymer that I mentioned, PLGA, stands for polylactic glycolic acid. It's a polyester, and that can be one polymer that you can work with. You can change that to albumin. There is just a few years ago, a chemotherapeutic drug packaged in albumin was approved by FDA. So you can change the polymer depending on what you want the release characteristics to be. And that polymer also can have different molecular weights, can be a copolymer. So you can change things around based on your needs. So we're not working with one single, with an element like you're working with carbon nanotubes. We're working with polymers to make these particles.
Other questions? Questions from the other sites? Join me again in thanking our speaker.
Thank you.